Hi everyone, I'm Bernadette and my group partners are Darren and Jonathan. And for our final group project, we decided to do Tay Sachs. So for the definition of the disorder, it's most commonly referred to or known as Tay Sachs, but other names include GM2 gangliosidosis type 1, B variant GM2 gangliosidosis, hexosaminidase A deficiency, and hex A deficiency. And there are three different forms which are distinguished by the age of onset for Tay Sachs, which include the infantile form, which occurs during the first few months of life, the juvenile form, which is childhood, or the late onset or adult form, which is late childhood to adulthood. For what happens to the body essentially fails to create either any or a sufficient amount of the beta hexosaminidase A enzyme, which aids in the breakdown of GM2 gangliosidosis. This is a fatty substance, and when there is a buildup of this substance, it leads to the various symptoms and eventually death. For the major signs and symptoms for the infantile form, symptoms include an exaggerated startle response to noise, loss of skills learned, which is regression, loss of muscle and mental functions, which leads to paralysis, unresponsiveness to their surroundings, blindness, seizures, and difficulty swallowing. And the most common cause of death for the infantile form is bronchopneumonia. For the major signs and symptoms for juvenile, there is frequent respiratory infections, behavioral problems, loss of muscle and mental function, seizures, loss of vision, loss of awareness, responsiveness to the surrounding environment, and their most common cause of death is actually respiratory infections. For the major signs and symptoms for the late onset / adult form, there is slow progression of neurological impairment, which can lead to clumsiness, muscle weakness, tremors, loss of coordination, etc. And they might need mobility assistance, and they can develop psychiatric disorders such as schizophrenia. And for the part of the body that it generally affects the central nervous system, which is the brain and spinal cord.
Tay Sachs is an autosomal recessive disease. Most notably, it miscodes the 15th chromosome. It requires two inactive genes or alleles for it to fully have its carrier be affected. So for example, if it's just one dominant and one recessive allele, it would be considered an unaffected carrier. However, if it's two recessive alleles, it would be a affected carrier. Those affected by Tay Sachs have an inhibition of the beta hexosaminidase A enzyme. This is caused by a deficiency in the GM2 activator, so therefore that it cannot actually produce these enzymes. These two characteristics cause the buildup of ganglioside lipids in the nerve and brain cells. While these lipids are accumulating, this actually destroys the nerve cells in the brain and spinal cord. Tay Sachs is categorized as a lysosomal storage disorder. The best description of these disorders is an accumulation of toxins which leads to cell death. In the case of Tay Sachs, this is caused by a buildup of lipids. In regards to gene expression regulation, the synthesis of the beta hexosaminidase A enzyme is either completely destroyed or has very little enzymatic activity. In the S phase of the cell cycle, the DNA synthesis checkpoint is missed in Tay Sachs. This causes the mutations of the hex A gene, most notably the proteins G269S and E482K are misfolded. These improper protein conformations cause it to be stuck inside the endoplasmic reticulum.
Okay, so for how the disorder is inherited, the specific gene responsible for this genetic disorder, Tay Sachs, is the hex A gene or the hexa gene. And the biological function of this gene, according to the Tay Sachs disease article, is the hexa gene provides instructions for making part of an enzyme mentioned earlier called beta hexosaminidase A, which plays a critical role in the brain and spinal cord. This enzyme is located in lysosomes, which are structures in cells that break down toxic substances and act as recycling centers. Within lysosomes, beta hexosaminidase A helps break down a fatty substance called GM2 ganglioside. It is not a sex-linked genetic disorder, and it is a recessive autosomal one. The particular chromosome that it's located on is chromosome 15, which can be seen in this karyotype. And lastly, an example of a pedigree for a person with Tay Sachs disease is shown right here. The black circles and rectangles, I know it's kind of hard to distinguish, they represent non-carriers, so they're homozygous dominant genotypes. And then the carriers are dark gray and they have a heterozygous genotype. And then on the bottom left, you can see an affected child which has a homozygous recessive genotype.
So how is this disorder treated? Unfortunately, there are no cures for this disorder. However, there are treatments that can help ease the symptoms for the average 4-year-old child with Tay Sachs. As this disorder targets the nerve cells and spinal cord, physical therapy is a critical treatment that can delay the deterioration of joints. Even simple exercises, such as stretching and moving, can be a great benefit. Additionally, anti-seizure and pain medication is recommended to ease the symptoms as well. Where there are no cure for this disorder, a preventive measure of gene therapy is currently under animal experimentation. Thankfully, so far there has been successes in which the conditions of mice with Tay Sachs were improved by introducing viral vectors that produced hexa enzymes. The increased levels of hexa would greatly reduce the GM2 gangliosides buildup and ultimately reduce the symptoms of Tay Sachs. If gene therapy is possible in humans, viral vectors will also be used to inject specific genes into the brain that would code for the missing hexa enzymes. How is Tay Sachs diagnosed? The Tay Sachs disorder is diagnosed through surface-level questionings regarding the children's symptoms and family history, and then a blood test is administered to confirm the disease. A blood test with low or absent levels of hexosaminidase indicates positive for Tay Sachs. Furthermore, an eye test may be administered to check for a red spot at the back of the eye, which is another indicator of the disease. Genetic counseling is available and recommended for future parents that may be concerned about the risks of their baby having the disease due to family history. In addition, enzyme screening is incredibly important if they suspect themselves to be a carrier of the Tay Sachs disease. This would provide the couple with the reproductive options and possibilities for a child with Tay Sachs. How many and what type of people are likely to have the disorder? This disorder is most commonly found in Jewish people of Eastern European and Russian descent. About 1 in 2,500 to 3,600 babies are born of Tay Sachs in those regions. Additionally, about 1 in 30 Jews of this origin are found to be carriers of the gene that causes Tay Sachs. Researchers believe that this is caused by the commonality of Ashkenazi Jewish ancestry. In comparison to the general population, about 1 in 320,000 babies are born of Tay Sachs. People of French Canadian, Pennsylvania Dutch, and Cajun descent are also at a higher risk of Tay Sachs.
The research paper our group chose is the reversibility of neuropathology in Tay Sachs-related diseases. This research article shows different treatment options for people that are suffering from LSDs. The first treatment option is a RAB injection. This study has only been approved for mice and is not yet in human trials. This injection is a plasmid injected into different parts of the brain at different times. The goal of this injection is to activate the beta-hexosaminidase enzyme as well as decrease the amount of lipid buildup. The timing of these injections is crucial. As you can see in the first graph, the sooner that this injection is placed into the body, the longer lifespan the mice will have. And graph B shows the same thing except with margins of error. Real-time PCR further emphasized timing in this experiment. mRNA extraction showed that the growth and activation of microglia and astroglia increases the disease process. Microglia and astroglia are just different neurons in the brain. As you can see by these two models of mice, the Sandler and Twitcher model, there are regional differences between the growth of these two types of neurons. So the sooner we can inject, the less time these neurons will have a chance to develop, and therefore the buildup of lipids will be lessened.